We're now going to move on to the presentation of the top 4 abstracts, and our first speaker is Megan Keiser from the Children's Hospital of Philadelphia, and she's going to talk about translating RNAi for Huntington's disease, intra-putaminal delivery of AAV21 MIHDS1 for comprehensive dosing, biodistribution, silencing, and safety in a non-human primate. Megan.
Thank you so much. That's my title. I want to thank the Society for allowing me to come here and share my exciting research with you guys. Here's my disclosure slide. So a little background on Huntington's disease. It's an autosomal dominant neurodegenerative disorder. It's caused by a CAG triplet expansion repeat within exon 1 of the Huntington gene. The mutation produces an expanded polyglutamine region in exon 1 of the Huntington gene. Here you can see from the cartoon, here's a normal Huntington exon 1. Regular people walk around with 36 or lower CAG repeats. Pathology begins to occur when the expansions reach 36 or above, where you have misfolding of protein. This causes a toxic gain of function and is ubiquitously expressed throughout the body. However, the brain is particularly susceptible to mutant Huntington toxicity, but other organs can be affected as well. Here I'd like to draw your attention to the slabs at the top. Right here is a non-affected normal person, and on the other side you can see the increase of the ventricle due to dying, really, of the putamen. So that's what a patient looks like. Onset usually occurs around 40 years of age, but is known to occur at any age. So from this, the mutant Huntington protein can cause cellular dysfunction, which manifests in several ways. One is chorea, or jerky, unwanted movements. There are behavioral and personality changes, and there's also a cognitive impairment. So instead of trying to focus on all of these downstream effects, we'll be using RNA interference to stop the mRNA and therefore knock down the mutant Huntington protein. So the Davidson lab has been working on this for about 15 years. We started at the University of Iowa looking at RNAi triggers in mice. We kept trying to perfect this technique, moving on into different mouse models showing efficacy, trying to optimize safety. Then one of our former postdocs went out to Oregon and works at the National Primate Research Center there and started a preliminary pilot study in primates. And then the Davidson lab migrated out east to the Children's Hospital of Philadelphia, and we've continued to work on safety and efficacy. So the study that I'm going to talk to you about today was completed at the Oregon Institute, looking predominantly at dosing and biodistribution of our therapeutic AAV21 MIHDS1 in the adult rhesus macaque putamen. This study used 12 animals, a mixture of male and females, all adults, that were bred and born at the Oregon Primate Center, and all animals, to minimize immune response, were screened for neutralizing antibodies, and only those that had less than 1 to 20 were included in the study. The doses are seen below. There was an N of 3 per group, mixed male and female. I'll just refer to them as the low titer, mid titer, high titer, and the diluent was vehicle. We targeted 2 areas in the putamen. The goal was really to cover the entirety of the putamen from rostral to caudal, and so we decided on targeting 2 individual sites within one hemisphere of the putamen. The pre-commissural putamen received 80 microliters, and the post-commissural putamen received 70 microliters. You can see here. We used convection-enhanced delivery, and we utilized a system known as ClearPoint, which is an intra-MRI neuronavigational surgical delivery platform that has been received and utilized by the FDA for, since 2010 in patients. Now the tower here is actually affixed to the patient's head while they're in the MRI during the entirety of the surgery. This allows you to make sure that your targeting is spot on and also to follow the distribution of your agent. This here is the same device that is used in patients that was used in primates in the MRI to aid in translatability from primates into clinic. So to be able to visualize what we were infusing, a lot of people spike the test article with a contrast agent, and we used gadateridol. And you can see here on the other side is at the beginning of the infusion here, and at the end of one of the infusion sites, 80 microliters, you can actually see that we're hitting our target quite well. We wanted to quantify this, and so using Osirix and a 3D slicer software, we looked at the gadateridol filling of the putamen throughout the diluent, low, medium, and high doses, and you can see from the full putaminal volume, we're hitting between 60 and 70% of the putamen, which we think was achieving what we wanted. So to make sure safety is a priority, the animals were tested at baseline and then every 2 weeks after surgery until necropsy at 6 weeks using a neurological rating scale that was optimized by Dr. McBride out in Oregon. And most importantly, there was 0% change in baseline in all groups, showing that there were no developmental or aberrant motor behaviors in any of the study animals caused by the intervention. We also equipped the animals with activity collars to measure their activity throughout day and night, and there was also no significant changes in motor activity or circadian rhythm. Then to measure very strictly the biodistribution, we took a prolific number of punches throughout the brain and surrounding tissues. Here you can see this is the putamen. We split the putamen into 4 4-millimeter thick slabs, A through D, and on the other side here, these circles were actual individual punches from each slab. There were 34 punches for each hemisphere of the putamen we collected, and then each individual punch was isolated for total RNA, and measurements were made from that analysis. So here's a cartoon roughly showing, here you can see slices or slabs A, B, C, and D moving rostral to caudal, and on the side is a color codation of silencing. Now we know from mouse studies that 40% silencing of endogenous Huntington can lead to efficacy in mouse models. And so here you can see that we're hitting very good coverage. The injection sites, just to know, we're about at B and C. Here there's quantification per slab and per treatment dose. You can see that we're getting significant knockdown in all groups throughout slabs A through C. However, in slab D, the most caudal region, the mid and high dose was also achieving significant silencing. On the other side, you can see on a punch-by-punch basis, each shape is a punch from the putamen. Each color is a different animal in the treatment groups, and you can see that we saw a very nice dose response here. Then we wanted, again, for safety, we wanted to look at markers for suppression tolerability in the injected putamen. And so we looked at DARP32, which is a measure for medium spiny neurons, IBA1, a marker for microglia, and GFAP, a marker for astroglia. And you can see that aside from some sparse outliers of individual punches within specific animals, it was very well tolerated. We also wanted to make sure that the therapeutic was specific to the central nervous system, and here looking at additional peripheral tissues, we didn't see any silencing of Huntington. So in conclusion, we found that this intra-MRI neurosurgery using an FDA-approved ClearPoint system was feasible and a practical approach to deliver our therapeutic AAV-MISD-HDS1 to the rhesus macaque putamen. Significant Huntington reduction was not associated with any dysfunction or weight loss in life measurements. Huntington mRNA suppression was not detected in any peripheral tissues analyzed, and most importantly, we identified 2 doses of AAV1 MIHDS1 that conferred significant Huntington mRNA reduction throughout the rostral to caudal extent of the putamen. That was the mid and the high dose. Further studies are ongoing in Oregon. We're using imaging, biofluid, and tissue collection to further assess safety and tolerability in longer-term studies. And I would like to thank the team, my mentor, Beverly Davidson, Alex Moss, that does a lot of this optimizing of the sequences, the clinical vector core, which made all of our therapeutic agent, and Johannes van der Leeuw is the director there, and then the Oregon National Primate Center, Jody McBride's lab, and funding from the NIH. And I take any questions now. Thank you.
Thanks, Megan. This talk is open for questions. Yes.
Yes.
Hi, that's really fascinating work. I've got a question. Once you knock down the mRNA, what happens to the protein? What's the half-life and turnover of the protein?
We've looked at protein in mouse studies, and the protein silencing is continuous as well. The microRNA is continuously expressed from the AAV induction, and so mRNA is just one measurement. I don't know the half-life of the protein turnover.
Okay.
But protein knockdown is maintained.
Do you know how quickly it occurs in the monkeys once you...
No.
Thanks.
And how long do your follow-up studies need to be to get sort of IND-enabling data for moving this to the clinic?
That's an excellent question. We think 6 months would be adequate for maintaining knockdown and safety, but we're willing to go out to a year if that's what they wish.
Okay. If there are no more questions, thank you very much.
Thank you.